Hello, my name is Tong Wu. I am currently a postdoc fellow with Professor Chuan He in the University of Chicago. I started the Cas-seq project when I was a graduate student, and now we are still working on its applications in different biological systems and also a single-cell version of Cas-seq. Cas-seq stands for kethoxal-assisted single-stranded DNA sequencing. Cas-seq is a technology that we can use a small molecule to capture all the single-stranded DNA species in the genome, which is mostly double-stranded. Kethoxal is known to bind guanines at the N1 and NG positions, which is exactly the Watson-Crick interface. This is very important because to map single-stranded DNA, we have to specifically map the opened Watson-Crick interface. And this is how kethoxal differs from all the other DNA and RNA labeling probes. We use kethoxal to selectively label and enrich those single-stranded DNA species from the whole genome, and then we sequence it with high-throughput sequencing techniques. So with Cas-seq, we can basically capture all the single-stranded DNA species on the genome. Those can be related with transcription or DNA damage or any other single-stranded DNA species you can think about. There's an enzyme called RNA polymerase or other transcription factors. They open the DNA helix and then they use one strand of the single-stranded DNA as the template to make RNA. So by mapping these single-stranded DNA bubbles, we can accurately measure transcription activities. Cas-seq can not only map active transcription on the gene coding areas, it can also identify a group of enhancers which process very high enhancer activity. In principle, Cas-seq can help to study all the single-stranded involving processes. Transcription is one of them, but also DNA damage and non-traditional DNA structures. Another thing is gene editing, which is very popular these days and also very promising technologies. For example, when people use a CRISPR-Cas9 to edit the DNA, there will be a single-stranded DNA bubble formed, so Cas-seq can also capture that. Another potential application of Cas-seq is that it can be helpful to find new biomarkers on enhancers, because many diseases are related to enhancer mutations, which may cause the activation or deactivation of an enhancer. Cas-seq, which integrate next generation sequencing, can be very powerful in understanding these questions.